I'll also be honored to present the tiragolumab plus atezolizumab randomized phase 2 trial for non-small cell lung cancer patients in the oral non-small cell lung cancer session. Tiragolumab is an anti-TIGIT antibody. TIGIT is another inhibitory checkpoint. It works in a similar way to PD-L1. When TIGIT binds to its ligand PVR on tumor cells, it suppresses the immune cell's ability to recognize the tumors. So when an anti-TIGIT antibody like tiragolumab blocks that interaction, that restores the anti-tumor immunity in the same way that blocking PD-L1, for example, with atezolizumab would do. We know from preclinical trials or preclinical work that cell lines and mouse models that are treated with the combination tiragolumab and atezolizumab had improved tumor control and survival over those treated with either antibody alone. And that was the rationale for first a phase 1 experience of the anti-TIGIT antibody tiragolumab. That'll be reported actually later this summer in the second virtual meeting of the AACR 2020. But suffice it to say, we know that this antibody is well tolerated and can be combined safely with atezolizumab. And so this was the background information for the CITYSCAPE a randomized phase 2 trial. There were 135 patients enrolled in this randomized phase 2 trial. They all had frontline non-small cell lung cancer without EGFR or ALK alterations. And they all had tumor tissue expressing PD-L1 at a level of 1% or higher as tested using the DACO 22C3 assay. That could be done locally or centrally. Patients were randomized to receive tiragolumab and atezolizumab or placebo and atezolizumab. And the primary endpoints were objective response and PFS. The results of CITYSCAPE showed that patients that were treated with the combination tiragolumab and atezolizumab had improved objective response and PFS over patients treated with placebo and atezolizumab. And in particular, patients who had high levels of PD-L1 expression on their tumor, more than 50%, had even more benefit from the combination as compared to placebo plus atezolizumab. But unlike other immune combinations that are available, these two drugs, when given together, were really well tolerated. There was very little difference in the side effect profile, combination experimental arm to placebo arm, with a slight increase in immune-mediated adverse events, such as pruritus and rash, arthritis, for example, and infusion reactions, but none so severe that it led to more patients discontinuing tiragolumab, for example. So this trial has led now to a randomized phase 3 trial to confirm its results. And that trial is ongoing. And if it is able to confirm the exciting results of this phase 2 trial, this will offer another chemotherapy-free option for our patients, which is exciting. The phase 1 trial of tiragolumab, just the dose escalation, and then the preliminary expansion in combination with atezolizumab will be reported